Hi, everyone, and thanks for joining us today, both in person and online. We really appreciate it. This is a UCD Citizen Science Community of Practice event, and we're really pleased that Annetta Zintl and her team are here today to talk to us about their network for insect vectors in Ireland. And Annetta is the coordinator of that. And just to say that in this context, vectors are mosquitoes, ticks, flies, fleas and lice, and we'll be hearing about that today. So I'm going to hand over to Annetta now. Oh, if you have questions, put in chat, please do, and we'll get to those at the end.
Hi, thank you very much. And thank you very, very much to Jane and Liz for organizing this event as part of the Citizen Science event, which really is a misnomer because I think this is the find the experts in the audience event. So I'm well aware that there are numerous people in the audience who know possibly more about the subject area than certainly I do. But what I wanted to do is just tell you a little bit about this project for which we got funding from the Department of Agriculture about a year and a half ago. And the project is called the Network for Insect Vectors in Ireland. And this event is really about what citizen science can contribute to the project. So these are the objectives of the project. Really they focus on biting midges and mosquitoes rather than any of the other vectors that Jane mentioned. So we are aiming to establish a network of expertise and or connect to networks of expertise. And I'm well aware that there may be very well people in the audience who are members of existing networks of vectors or insects or arthropods that are involved in disease transmission. And then to carry out surveys of biting midges and mosquitoes. The other objectives relate to identifying the potential pathogens these vectors may be carrying, the hosts that are important for these vectors. Then there are some objectives that are aimed at finding new ways of identifying large numbers of vectors rapidly, because it's a very labor-intensive method if you do it morphologically or using the traditional methods. And then there is one objective which relates to developing teaching and learning material. But really, it's the first two objectives that I think are relevant for the event today. So very briefly, before I tell you a little bit more about the project itself, this year is the team that is involved. So the project is led by myself and Denise O'Mara, who is in the Southeastern Technological University. Then we have collaborators in the National Museum of Ireland, Aidan O'Hanlon, and in the department itself, James O'Shaughnessy. But the people really who are doing all the work are in bold here. So this is Angela Valcarcel Olmeda, who is here. She is our postgrad in UCD. Thomas Curran, who is here as well, postdoc in situ. And Elsie is here, who is also postgrad in situ. So they are the three people who are doing most of the work on the project. So as far as the objectives are concerned, so as you will remember, the first objective is about creating or coordinating a network of expertise. And the main sort of method that we have that we're using for this is to organize these quarterly webinars. And here are just some of the previous speakers that we've had in the last couple of occasions. The next webinar is actually going to be, is already scheduled for July. And it's one of Elsie's colleagues from Kenya, who is currently working in the United States, who is going to give us more of an idea of midges and mosquito-borne disease in the non-European context, which I think will be very interesting. But to keep this very short, just to let you know, please let me know if you want to be on the mailing list for this webinar. These webinars, you're more than welcome. Just send me an email, or indeed, if you feel you would like to present at one of the webinars, if you're working on midges or mosquitoes, please let me know, and I'd be delighted to have you included as a speaker. But in terms of the surveys that we have actually been doing for midges and mosquitoes, just to tell you a little bit what we have been doing so far. So obviously, when you're trying to catch these little creatures, it's important to know how they live, where they spend most of their lives. And with regard to biting midges, and I'm preaching to the choir here, I'm apologies if this is all too familiar to you. But what biting midges do is that they oviposit, so they lay their eggs into very organic, rich organic soil, wet, rich organic soil. So, you know, the area, I realize the pointer doesn't work on the screen, into these, you know, muddy areas, especially around drinking troughs, for example. And then the larvae hatch, and the larvae are free living, so they are not parasitic. They go through several molds and eventually they pupate and the adults emerge. Both males and females actually can live on plant nectar, but females have to feed on blood in order to produce eggs, in order to have the protein to produce eggs. Now, midges, as we probably all can testify, if we have been anywhere in areas that are plagued by midges, have very low host specificity. So they're very happy to feed on anything and anyone who comes along. They're very poor flyers. They are active at any time of day or night, mostly if the weather is calm. So they're not very good at flying in. If there's any windy conditions, they stay on the ground. And they live mostly outdoors. Now, people used to think that they could protect animals from midges by keeping them indoors, but that doesn't work very well. They will come in if there are hosts there. So what we've been doing in terms of collecting midges for our surveys is we have been setting traps, which are called on-the-support or OVI traps. Which I've actually got one of them here, but we had no hooks to hang them from, so it's still in the box. The way these work is that they are hung overnight in a stable or near animals, near livestock. And you can see there's a little power cable there, so it's plugged in and there's a blue light comes on and the midges are attracted to the light. There's a net around the light which prevents larger insects from flying in, so you don't have a huge bycatch. And the smaller midges get through the netting and they're sucked in then by a fan in the top of this net here, sucked into the funnel and down into a container that contains water with washing up liquid. And that's where they get trapped. And then in the morning, you can pick up the container and we strain it then and pour the midges straight into 70% ethanol for preservation. So that has been going on. The midge collecting was carried out for 18 months in 13 sites around the country, every 2 weeks for one night. So this was done by an incredible network of colleagues and friends and acquaintances. Anyone who even expressed the slightest bit of interest, we swooped and asked them would they do this for us. So it was really incredible. Many colleagues from the Department of Agriculture who have been collecting samples for us. So we've now completed the midge collection. We don't want any more midges. Certainly Angela doesn't want any more midges because she has been sorting through the catch. And the catch is there is a lot of work in that, sorting out even just a bycatch, the many flies that aren't biting midges, and then to try and identify the biting midges morphologically, and eventually also using DNA. So for the midges, we're pretty much, you know, we have all the samples that we want. For the mosquitoes, not so much. So again, mosquitoes, to know how to catch them best, it's important to know how they live. They also, the adult females also lay their eggs in or near water bodies, so really in stagnant water bodies. And the little larvae, they emerge and again go through several molts, pupate, and eventually the adults emerge. Now, the thing about mosquitoes is that they really can't breathe in the water. So they need to have a connection to the air outside the water. And that's where they have these little breathing tubes here, which they use to breathe. And they hang upside down at the surface of the water and use these breathing tubes to breathe. And as a result of this particular way of breathing, they can really only live in stagnant water. If there's any turbulence in the water, they're pushed under the surface too often, so they can't survive in water that has any sort of turbulence in it. The only females feed on blood, not the males. There are species that are very much focused on feeding on humans. Others feed on any mammal pretty much. Some species particularly like to feed on birds. Also, the types where the places where they leave their eggs are very species specific. So certain species like tree holes of all things, some species like car tires. Car tires are a fantastic source of mosquito larvae. So there are often plenty of them lying around. So that's where they like to deposit. Some of them deposit their eggs. Some mosquito species also like to come into human habitations, so they even spend time in between feeding and egg deposition inside dwellings. So these would be endophilic species. Others, the exophilic species, spend most of their life out of doors. Most mosquitoes like to, and midges as well, really like the sort of twilight times of the day, but there are some species that are, other than that, are diurnal, that is, they are active during the day, and others that are active during the night. Now, we did try to collect mosquitoes the same way as we're collecting midges, by setting up traps that would collect adult species, but we were really, really unsuccessful doing this. The problem is that for these traps, to attract mosquitoes, you really have to have some sort of a lure, something that they can smell. So both midges and mosquitoes are attracted by CO2, but you have to, for mosquitoes, you really have to have quite a strong source of CO2. We had this yeast culture, which smelled lovely, but it wasn't very effective in collecting the mosquitoes. And what we ended up with was just a very high bycatch, and we were worried we were actually depleting the biodiversity without getting very many mosquitoes. So what we have decided to do for mosquitoes is really go for these immature stages, because the beauty of this is that you can go, you can go to one site and you can look for mosquitoes. You can see them really, really easily. And Angela will show you some videos. We also have brought some live mosquito larvae. Unfortunately, it has been too cold and it's too early in the year to have nice big ones. But if they are big, they're so easy to see. So even I can see them. And if you have any sort of stagnant water anywhere, your garden pond, or like I say, these piles of tires that are very commonly found in the countryside, people use them for silage making and so on. So you will find mosquito larvae in them very, very easily, and any lay person can see them very easily. So that's what we've decided to focus on. And in fact, last summer, we went in a transect across the country. We started here in the west and went across to the east. And the beauty of it was that you didn't have to set the trap and then come back after a night or so. We literally drove along and hopped out of the car and did some collected samples along the way, and you can literally you pick out your mosquitoes. You take a scoop of water, put it into your white tray, and you can see the larvae very, very easily. And there was a lot of interest from all the farming community who followed very closely where we were at any point in time. So that was quite funny. But in addition to the immature stages, the larvae and pupae that we collected, we also collected some eDNA sample, which some of you may be working with eDNA as well. This was specifically to determine whether in sites where we didn't actually see any larvae, whether they are still there, but we just couldn't see them at that point in time when we went to visit the site. And we are planning now this summer to do another transect across the country, probably going along the west coast from north to south. So how can citizen science be involved in this or help with this research? Very, very obviously, particularly looking at the audience that we have here, is the expertise, the local expertise, the experience of many colleagues here in UCD. And I know, because several people have contacted me already, that there are numerous people in the audience who know a lot about water invertebrates and or various different kinds of insects or groups of insects. So certainly if we can connect and benefit of each other's expertise, I think that would really be beneficial for everyone. In addition, if anyone feels so inclined, we would be absolutely delighted if you wanted to collect some mosquito larvae as well and send them to us, or indeed hand them to me here on campus. We have some very opportunistically and optimistically made up some sampling collection kits already. So you're very welcome to take some with you. And like I say, Angela is going to explain in a minute how the samples are being collected. And like I say, it's incredibly easy. And once you start, it's actually quite difficult to stop. Every time you go past a water puddle, you keep thinking, oh, there might be something in there. So this is from our last field trip there, where we were in James's farm, and his little cats were very interested in what we're doing there. So other than that, like I say, if you're collecting specimens, if you're collecting freshwater invertebrates, or indeed mosquitoes as part of any other project such as BioBlitz, please let us know. It would be always really nice to join forces on this. If you are interested in helping with sample collection, even one sample, we're delighted. It's another point on the map. It's negative. We're also interested in negative sites. So any kind of sample, if you feel so inclined, we'd be absolutely delighted. Then in terms of adult stages, this is also very interesting. So across Europe, there's now a big concern because certain exotic mosquito species have started to appear in Europe. And they're certainly moving further onto the continent. And as Aidan O'Hanlon pointed out to me, it's the citizens that detect these exotic species first. It's never the people who go out into the field, because you'd have to have too many boots on the ground. The exotic species are really characteristic. They're actually very easy to identify, and Thomas is going to tell you more about this. So if you do if you do see something, and especially after today, after being pointed to what to look out for, if you see anything, please shout. Everyone in the country would be very interested to know if someone saw any of these mosquitoes here. So you can send them to us. You can send them to the lovely lady in the Irish Times, or a picture rather than a mosquito, I imagine, who does the identification of things that are found. What's her name? Anya? Yes, that's right. Or you can also send them to us, of course. We'll send them on to Aidan O'Hanlon, who is really the expert in identification of mosquitoes. But there are also some really nice online tools. So there is a mosquito alert tool. It's an app where you can also submit images. And then there is this reverse identification key for invasive mosquito species. And I have a hard copy of it printed out there. So you can have a look at this as well. But they are very, very distinctive, these exotic mosquito species. So I think if that's what people took away from this, would be already a huge achievement. Here's our project website. It does look very amateurish, even if I say so myself. but we do try to put up whatever materials that we're developing in terms of sampling protocols or so on, identification protocols, we put them up there. And we also always have information about our presentations, our upcoming webinars on the website. So without any further ado now, I want to introduce the next two speakers. So the first is Thomas, and he's going to tell you a little bit more about the adult mosquitoes and invasive species. And after that, Angela will tell you how to collect and identify the little mosquito larvae.
Thanks, Annetta. Oh, sorry. So thanks all to everyone here and online for taking the time out of your day to join us to hear about insects, well, mosquitoes and midges in Ireland. So today I'm just going to talk about how we can morphologically identify and differentiate mosquitoes from other insect groups in Ireland and really hone in on those invasive species that Annetta was talking about. So mosquito monitoring is obviously highly important in Ireland and globally because of the burden that they pose with various different diseases and the burden that we see already globally through different mosquito-borne outbreaks. For example, in South America this year alone, there's already been over 1 million recorded cases of dengue. And in Spain, we can see more vector-borne diseases coming out on the rise and making their own presence known. So in Ireland, we have recorded by the late Paddy Ash 18 species of mosquito in Ireland, but this contrasts that of the UK, where to date we know that there's 36 species existing in the UK. So we have to increase our surveillance efforts so that we know what exactly is here and how we can mitigate any potential risks. So mosquitoes have a very typical and general life cycle. So adults will lay an egg in a preferred habitat, typically stagnant water or tree holes or moist soil. When eggs are then oviposited, they can mature into larvae. There's 4 stages of the larval development, and these are also called instars, and it's just based on the growth and the size of the larva. Then post that, it'll form a pupa, and then it'll stay as a pupa for only a couple of days before it emerges as a so the egg, larva, and pupal stages are all water-based, and then following that, it emerges into a completely terrestrial mosquito adult. So we have mosquitoes in Ireland, but how can we actually tell which is a mosquito and which isn't? Oftentimes when we're out and about and we think we have a mosquito, we swat it away faster than we can actually see what's coming for us. So mosquitoes often get mixed up with chironomids, but chironomids are typically a lot smaller and their wings are typically larger than their body. Smaller than their body. The other one is fungus gnats. Fungus gnats look incredibly similar to mosquitoes and they're a very similar size, so it's hard to differentiate the two insect groups. But fungus gnats have much larger antennae and the females don't have a proboscis, so they actually can't feed off of humans or bite. And the other one is a crane fly. Crane flies are a lot larger than mosquitoes. They're like kind of slender, spindly legs is often what gets them confused with mosquitoes. But again, they don't have a proboscis, so they can't actually bite humans or animals. There's 3 main characteristic features of a mosquito. So the first is called the maxillary palps, which are 2 kind of appendages on either side of the mouthpiece. You have the cerci, which is part of the tip of the abdomen, which is involved for the development and laying of eggs. And characteristic of mosquitoes, particularly females, is their proboscis. So in Ireland, we have 5 different, oh, sorry, I forgot about that. This was the next slide. So these are our typical mosquito characteristics, other than those 3 identifiable features I mentioned before. So their size ranges tends to be between 1/4 to 3/8 of an inch. Their body is narrow and oval shaped, so you'll often see that on any kind of graphic of mosquitoes. Even when they're sitting, they kind of form this oval kind of resting position. They're typically grey, but there is still some slight colour variation. For example, the most dominant species of mosquito in Ireland that we've come across and other people have come across is Culex pipiens, which are light gray in color, but on their abdomen have these kind of pale white stripes that go across it. But it's a sibling species, Culex fasciatus, also light gray in color, but instead of having these kind of stripes or pale white stripes on its abdomen, it's just completely pale. So have long legs, and females have a proboscis or a long nose for biting and feeding. So in Ireland, we have 5 different genera of mosquitoes, but I'm only going to go through typical characteristics of the main 4 you're likely to encounter. The first is Aedes. So Aedes have a very pointed abdomen, and it's very characteristics, especially of Aedes cantans. So they generally have black and white bodies and stripes on their legs. You can see that from a mile away, especially for some of these invasive Aedes species, which we'll get on to soon. The next is Anopheles, which, unlike other mosquito species, their legs are a lot more kind of longer and spindly-like. They have long palps, which again are those like mouthpieces kind of at the mouth. And they have spotted wings as opposed to any sort of venation and or clear. Next, we have Culex. So Culex are dark brown to black, and they have a rounded abdomen as opposed to that pointed abdomen that Aedes tend to exhibit. And again, only in females, they have a very pointed proboscis, so watch out for those. And lastly, I'm going to talk about Culiceta. So this genre of mosquitoes are much larger typically than your other species of or other genus of mosquitoes. And I don't know if you can really see it, but they have a very long drooping proboscis. So other species of mosquitoes, their proboscis is just kind of straight out and they kind of dip their head to get in, whereas on Culiceta, it kind of droops at the end. But the European Centre for Disease Prevention and Control have identified a need for mosquito monitoring, particularly for 6 different Aedes species, because of their progressive movement from non-endemic countries all throughout Europe and because of the massive disease burden these species exhibit. I'm only going to talk about 3 of the main species that are really being monitored by researchers throughout Europe because of their presence across many different European countries. So Aedes aegypti, Aedes albopictus, and Aedes japonicus. So the first thing that we look for, and to me, I find it the most identifiable feature, is their head. So for Aedes aegypti, you'll see that they have silver scales throughout the head of the species. And if you look closely, it exhibits this kind of shape of, if there's any musicians in the online or in person, it exhibits the shape of a lyre, which is very similar to kind of like a harp kind of on its side, but a lot smaller. In the middle, we have Aedes albopictus, which tends to typically just have a straight silver line down through its headpiece. And most definitive is Aedes japonicus, which is the only one I've come across that seems to have these golden scales on its headpiece, but it'll have 5 golden lines. The next is the body. So Aedes aegypti will have these pale lateral lines going across, and then these thin basal lines down its side. Aedes aegypti. Aedes albopictus, again, has these thin basal lines, but at the side, they're more elongated than Aedes aegypti. And then Aedes japonicus will have pale lateral and middle patches. So that's the difference between that. Rather than being this sort of line formation, it's just patches across its body. And then the last part are its legs. So when we talk about trying to identify different mosquito species from their legs, we're looking at the different segments throughout their legs. So for Aedes aegypti and Aedes albopictus, the 5th segments, which are just here at the bottom, are entirely white. So if you're only going based on legs, good luck. You're going to need the other pieces as well. Whereas for Aedes japonicus, the 4th and 5th segments are black. So for that, you can differentiate it anyhow. But what we're here to talk about is how the public can assist us in our mosquito efforts. And I'm bringing it down to just 3 points. Firstly, notification. Notify us if you come across any mosquitoes or you think you have a mosquito. We're happy to collect it. We're happy to do more work on it. Or if you're concerned about whether or not what you have is a mosquito or not, we do have the reverse identification key available as a QR on our website. So do check that out and increase our foot traffic. And secondly is the collection and the submission of the mosquito samples. If you are going to collect them, you can get in contact with any of us on the research team. We can give you an address to send it to, or if it's close proximity, I'm sure none of us will have any issue going to actually collect it for you. That's no bother at all. And lastly, as Annette has already described, cooperation. If any of you elicit any sort of minor interest in the project, we'll be on to you. So that's everything I have to say. Next up is Angela.
Hi, thank you all for coming, and thank you also to the online audience. I'm just going to talk a bit about how do we collect mosquito larvae and how would you, if you want to collaborate with us, should do it. So we have already spoken about the life cycle of a mosquito. So if we're looking for immature stages as larvae or pupae, we are going to look for them in water bodies. As mentioned before, the type of water body really depends on the species, but mainly stagnant water bodies are the ones that we will be focusing on. I've got a video here. I hope it's, yeah. Okay, so this is a drinker that wasn't being used at the moment in a farm. And I just stopped by and saw all these hundreds of larvae on them. As you can see, all these like lines there are mosquito larvae. They're just chilling there, breathing. But whenever they can feel the presence, they can see you. So they would try to just get to the bottom of the container or the feature or whatever they are on to escape from you. Oh, yeah. So we have already seen images, but I just want to get a few characteristics of the morphology. So the body is divided as well as the adult in three main parts. So the head, the thorax, and the abdomen. But the main part that is characteristic for these insects or these instars is the siphon, which is the one that they use to breathe, and that would be stick to the surface of the water. And you can really distinguish mosquito larvae from other insects because they have this Y-shaped tail, let's say. Larva behavior is also really interesting when you are trying to collect it, because they will tend to rest upside down on the surface of the water, as you can see in that falcon tube there. Their size can vary between instars and species, but they tend to be between 0.5 and 1.5 centimeters average. They have this wiggly swimming, so they're very mobile. They will be, if they are disturbed, they will be just swimming around, and it's really characteristic how they move. And again, if danger is detected, they will quickly swim to the depths, trying to run away from it. About pupae, we have already seen some pictures. They are comb-shaped. They're a bit bigger than the larvae, mainly, and they are more static. However, they can swim too. They are a bit more difficult to collect because, as Thomas said, they only spend a few days being pupae, while they can spend about a month being larvae between the 4 different instars. So how is our procedure to collect mosquito larvae and pupae? The first thing we will do is look for a stagnant water body. It can be any water body that you find around. It can be a bucket with rainwater, or it can be a drinker, as we saw previously. All tires are fantastic collecting rainwater because of the shape, the water cannot escape. So if the weather, like if the temperature is not really high, it won't evaporate either. So it's amazing for mosquito breeding. And then also other, let's say, more temporal, yes, sorry, thank you, Annetta. Temporal places like ditches or puddles that might seem that they dry out in a few days, but here in Ireland, where the weather is so wet and it rains every other day, they usually stay long enough for larvae to develop. Then we will scoop some water from the surface. You have to do this relatively quickly so they don't see you and run away from you. And then we put the water on a white background and let it settle so the larvae calm down and come to the surface of the water on the tray. Just like that. It's very straightforward. Then we will look for mosquito larvae, as we have mentioned before. The morphology of the larvae, the way they move is really was key here. And then we will collect it with a pipette and we will place it in a sealable container. It can be any sealable container. They are pretty happy, whatever, actually. So you can see here's some example of how it's done, how we actually did it on our field trip this July. So you just pick them out with a pipette. It's a bit tricky at the beginning because they try to run away from you. But once you've done it a few times, it's actually challenging and nice. You just want to keep collecting larvae. And yeah, and some pictures of how how you will see it actually on the field. Finally, filling out the recording sheet is essential for us because we want to know how the surroundings were, how the weather was, the temperature, everything. The more information we can get, the better for us because we can picture better the place where you actually collected the larvae. Even pictures are welcome, of course, everything you can think of. And then you can store the sample at 4 degrees. So that's in the fridge, and contact us so we can collect the larvae or tell you where you can send it, whatever. So thank you so much. If you want to visit our website, this QR will just lead you to the NetVec project website, where you have all the information, you have all the documents that I mentioned. So there's the recording sheet, there's also a flyer explaining all these bullet points, and there's also a video that you can have in your mobile phone or whatever, in case you just bump on a water body and want to do this, you have all the information there.